Hi guys, welcome to your next installment of video notes for IB Bio. Morpheus would like to say hi Morpheus, hi class. So today's notes are for 6.3, Defense Against Infectious Disease. So microbes that cause disease are called pathogens, physical barriers that we have in our body against pathogens. You have two main categories externally or physically, and that is your mucous membranes and your skin. So you can see mucous membranes down on the left here. So mucous membranes produce a sticky mucus. That mucus is there to trap invaders. The pH of that mucus is not favorable to pathogens, and you have this mucus in your nasal cavity, in your mouth, and also in your genitalia region. There are also enzymes in these areas in the mucus that helps to break down pathogens, and those enzymes are called lysosomes. And there are also natural organisms that live in these mucous membranes. And these work by competitive exclusion by the non-harmful microbes, so they compete with the harmful microbes. And then, of course, you have your skin. This is commonly thought of. It is continuous. It is hard to find an opening. Any openings in your body are going to have mucous membranes, but it's not going to be a puncture in the skin. There's many layers to your skin. It's very tough. Your skin is very dry. There is a pH associated with your skin, and that pH is not favorable to pathogens. There are also lysosomes on your skin, not in as much quantity as in the mucous membranes and in the sticky mucus, but there are some. And there are natural organisms that live in your skin or on your skin as well. All right, so the first thing we want to talk about is what if your skin gets penetrated. So that would be a cut, and what happens. So the process that happens is called blood clotting to seal the cut, and this is going to seal the wound and prevent a further loss of blood and blood pressure. This involves actually a cascade of reactions, each producing catalysts for the next reaction. So if we look to the right here, you have a break in the skin barrier, and then platelets will go to the cut or the break, and they will release molecules called clotting factors. Those clotting factors then trigger production of a molecule called thrombin. Thrombin will then catalyze a molecule called fibrinogen into fibrin. Fibrin now forms a mesh that will trap more platelets and will trap the blood cells. And so that is actually, the fibrin is what is going to form your scab. And that is what's going to dry out and form the scab. All right, so now that we're talking about, we have talked about the heart in the last lecture, and an issue now that we're talking about blood clotting is coronary heart disease. And that is also called atherosclerosis, and this can lead to blood clots. And if these clots occur in the myocardial tissue or the heart, we call it coronary heart disease. And a heart attack, also known as a myocardial infarction. So if you watch any medical shows, that is going to be what the terminology they use, or if you're at the hospital for a heart attack, that occurs if a coronary artery becomes completely blocked. So that is one of the reasons the coronary artery is the main artery, if you remember, that supplies the heart tissue with oxygenated blood. And if you remember, we did also point that out and look at that during our heart section. So coronary muscle tissue will die as a result of the lack of blood and oxygen to it. All right, so let's continue to talk about coronary heart disease. There are some risk factors. One is that some people, it's genetic, are predisposed for high cholesterol levels or high blood pressure, and this is going to lead to an increased risk in coronary heart disease. Older people are typically at greater risk because their arteries are less elastic, and then males are at greater risk than females. People who smoke, the smoking constricts the blood vessels, and so this is going to increase blood pressure, heart rate. It's going to have a decrease in oxygenation of the heart muscle and an increased fibrinogen and platelets in the blood, which will lead to more clotting. A diet, of course, increases in fat, cholesterol in the blood. This is going to lead to plaque formation in the arteries, and then that could lead to coronary heart disease. And exercise, lack of exercise is going to lead to an increased risk because you're going to have a weakened circulation pattern. Obesity, just like diet and exercise, being obese is going to increase your blood pressure, and this could also lead to a plaque formation in the arteries if you are obese. And then stress. Stress has actually been linked to increased cortisol hormones in the blood, and those increased cortisol hormones can actually cause atherosclerosis as well. All right, so moving on, I know this seems like we're talking about the blood a lot, but the blood is important in infectious disease. So we have some specific blood cells. You've referred to them as just white blood cells, but their actual name is leukocytes, leuko referring to white, and then cells in the blood. And there are many different types of leukocytes. The main ones that we're going to talk about are lymphocytes and phagocytes. Phagocytes are typically the vacuum cleaners. There are different types of those as well, one being called a neutrophil. Those are ones that attack bacteria and fungi. You also have leukocytes that we're not going to talk about this year, eosinophils, basophils. So eosinophils are ones that really work towards parasites. Basophils are ones that are, basophils are ones that are indicative with our allergies. But our phagocytes are vacuum cleaners, and they actually help and work with our lymphocytes. And lymphocytes include 3 different types of cells called B cells, T cells, and killer cells. So we'll get to those in a little bit. All right, so talking about the leukocytes and the phagocytes. So if a pathogen enters the body, the first line of defense that we have is something called a phagocytic or phagocyte leukocyte. So it's a particular type of leukocyte, phagocytic leukocyte. Phago, of course, if you remember from endo and exocytosis, phago means eating. Cytic, of course, refers to cells. Leuko, white, and then cytes refers to blood cells. And so this video, hopefully we can get this neutrophil in the warp path. So this is just a quick little video. You can see this large thing moving. That's actually a neutrophil. Those are other blood cells within the blood. It's chasing a very small bacteria. And clearly not in the body, but in a microscope. So it is trying to chase that sucker down, and he has now eaten him or ingested him. And this is a little cartoon of what we just saw with the neutrophil. So we click on the play button, and we're going to see how white blood cells deal with a bacterium. So the white blood cell, it's attracted to a place that's been infected. So this is our infection bacterium, and then it engulfs the bacterium. Then once it's engulfed it, enzymes from the cell digest the invader, and then the soluble products are absorbed. So we'll watch it one more time. So this phagocyte, he does phagocytosis to eat the bacteria cells that are in the blood. All right, so let's talk about this phagocytosis. So phagocytosis and phagocytes are also called macrophages, lots of different names in this unit. Macrophages engulf the foreign bodies like we just saw. They detect and move towards particles by chemotaxis or chemical movement. They flow over the particle, they engulf it, and then once the particle or the bacterium is inside of the macrophage or phagocytic leukocyte, then it's going to have it lysosomes released inside of it, and those lysosomes are going to release lysozymes, which are enzymes that will kill and digest those microbes. So you may not know, but when you get an infection and you get pus, That is because phagocytes have gone to the infection. And so what your pus is actually dead, fallen phagocytes or macrophages. I have two quick little videos to help us with these concepts.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis. Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles such as old mitochondria. This results in the destruction and recycling of these structures.
All right, so that gives you an idea of how those lysosomes work. All right, so now the first video about the macrophages did talk about antigens. So let's talk about what these antibodies and antigens are. So antigens are just proteins that are embedded on the outer surface of a cell or a virus. And so any chemical that stimulates an immune response is going to have an antigen on it. Okay, so you can see here in this red picture, we have two different shaped antigens, and so they will interact with the other particle called an antibody differently. So an antibody is a globular protein. It is shaped similarly to a Y, and it is produced as part of the immune response. They are specific to the antigen on the pathogen, and the antigen binding site is analogous to the active site of an enzyme. So the antibody and antigen work together like a lock and a key, but not necessarily, not to produce a product or to break down a substance. Instead, this is how our body will attack a foreign molecule. All right, so quickly let's talk about viruses versus bacteria because they're and review these concepts. So viruses are very small, bacteria are much larger. The viruses do not have a cell wall, but they do have a protein coat. Bacteria, on the other hand, do have a cell wall. Viruses don't typically have DNA. They typically have RNA. Bacteria does have DNA. It's in loops, and it does have that single circular plasmid as well. There is no nucleus in either. In the viruses and in the bacteria, it is free-floating. In the bacteria, the DNA is typically found in a region called the nucleoid, but it's not an enclosed area. And then the cell surface, some viruses have, or most viruses have spikes or attachment surfaces for invasion into a cell. Bacteria, though, typically just have flagella or cilia or even pili for exchanging chromosome information or plasmids with another bacteria set. So big key here is that if we have a virus infection, we don't want to take an antibiotic, and we'll get into this more later, but really most antibiotics work on things mainly such as this cell wall. So since viruses don't have a cell wall, the antibiotic is not going to work towards it. So let's talk about how these antibiotics work.
How do antibiotics work? Pathogenic bacteria in the body cause infections, which can be treated by antibiotics. Antibiotics can be bacteriostatic or bactericidal. Bacteriostatic antibiotics slow the growth of bacteria by interfering with the processes the bacteria need to multiply. These processes include DNA replication, metabolism, e.g. enzyme activity, protein production, bactericidal antibiotics kill the bacteria, for example by preventing the bacteria from making a cell wall. Penicillins are bactericidal. Penicillins include penicillin 5 for sore throats, amoxicillin for chest infections, and fluxloxacillin for skin infections. Antibiotics can be so-called broad spectrum, affecting many different bacteria in your body, including useful bacteria in your gut. Some antibiotics are more narrow spectrum, only affecting 1 or 2 types of bacteria. It is better to use narrow spectrum antibiotics where possible. Most antibiotics have no effect on your immune system. Antibiotics do not work on viruses because viruses have a different structure to bacteria. Viruses incorporate themselves into a host cell in your body in order to multiply. Bacteriostatic antibiotics that affect bacterial DNA, metabolism, or protein production do not attack body cells and therefore do not slow the growth of viruses. Viruses do not have a cell wall, and therefore bactericidal antibiotics that act on cell walls cannot kill viruses.
All right, so how do antibiotics work? They will, as the video said, stop DNA replication. They will attack the cell wall and membrane. They could stop transcription from happening, or they could stop translation. Also, as the video stated, there are 2 different types of antibiotics. They both work on bacteria, so the prefix to the names are bacterio. So bactericide is a bacterial killer, and bacteriostatic is a bacteria staller. So if you have an infection, so you're going along fine, then you get an infection, you start to have signs and symptoms, so you go to the doctor, he or she determines it's a bacterial infection, so they give you a bactericide, and then that's going to kill the bacteria, and we have this immediate drop, and then the macrophages within your body will help clean up, what's left of the infection. Same type of thing. You go to the doctor, you get an infection, you're living life, and then you get an infection. You start to have signs and symptoms, so you go to the doctor. He or she determines it's a bacterial infection, but this time instead of giving you a bactericide, they give you a bacteriostatic. And what this does is it stops the growth, so we have a plateau. And it's okay, you will get you will get better. You still have this decrease. It just takes some time because we're going to let our own lymphocytes of our body kill the pathogens that are left, and then our macrophages, will clean up the infection. All right, so antibiotics are really ineffective against viruses. The overuse of antibiotics is accelerating the evolution of more harmful bacteria. We are running out of antibiotics that work and are selecting for diseases such as MRSA. MRSA stands for methicillin resistant Staphylococcus aureus. Methicillin is a type of antibiotic, and so having a Staphylococcus or a bacteria that is resistant to methicillins is a really big deal. Here is just another area, another visual cartoon of what the antibiotics are going to work against. So antibiotics are designed to disrupt structures and metabolic pathways, as we already discovered, and they do not work on viruses because viruses do not have these pathways or these structures. So this is a universal antibiotic that's going to stop the mRNA progressing into the ribosome. This is tetracycline. You might have heard of that one before. That one actually prevents the tRNA from binding at the A site during translation. Then we have erythromycin, also might have heard of that one. That's going to interfere with how the protein progresses because it's going to stop the tRNAs from being able to exit the ribosome. Clindamycin, that's going to stop the peptide bond from being able to form. So again, we're not going to have any proteins that are made. And this one is going to stop the tunnel passage. So it's going to prevent any of the amino acid polypeptide strand from getting out. So that animation showed how the Nobel Prize was won in chemistry in 2009. So again, why don't antibiotics work against viruses? So first of all, viruses are a host, and they use a host cell metabolism. They're not hosts, They use host cell metabolism. So they inject their genetic pieces into a host cell, and then the viruses are protected by the host cell. So you can still kill the host cell, but those viruses are still in your body. Because viruses have different metabolic pathways than bacteria, the antibiotics are not going to work on them because they have different structures. They're not going to work on them. So we have a problem when we have indiscriminate use of antibiotics. If you get sick and you go to the doctor and your doctor tells you you have a virus and you say, I just want some medicine to feel better, and they give you an antibiotic, that is not going to make you feel better because you don't have a bacterial infection. You may think that you feel better because perhaps your body is able to fight against the virus, and so you have a correlation of a reduce in your signs and symptoms while you're taking the antibiotic. Antibiotics become ineffective against bacteria as we continue to use them. And this is an example of evolution by natural selection. So over many generations, multiple resistant strains of bacteria can develop. And so there's some information about MRSA. I'm going to do a little lesson with you in class about that. And that is a big superbug that was found first in hospitals.
Bacteria are becoming resistant to many of the antibiotics in our medical arsenal. One reason for the widespread resistance lies in the indiscriminate use of antibiotics. For example, at the insistence of patients, doctors often prescribe antibiotics even for the common cold. Antibiotics have no effect on viruses, and viruses cause most colds. Antibiotics attack bacteria. There are millions of bacteria in the body. Most are found in the gut and are either harmless or beneficial. Some of these bacteria are beneficial because they make vitamins that the body can use. Also, by competing for space and nutrients, the many species of bacteria prevent each other, as well as other harmful bacteria, from multiplying out of control. What happens to the resident bacteria when a person takes antibiotics? If the person has never taken this particular antibiotic before, then most of the bacteria would probably be susceptible to it. Antibiotics work by a variety of mechanisms. Tetracycline, for example, binds to ribosomes in bacteria and jams the production of proteins. From lack of protein production, a bacterium dies. Most of the bacteria die, but by chance, a few may have mutations that allow them to live in the presence of the antibiotic. Their mutations might cause their ribosomes to have a slightly different structure, which prevents the antibiotic from binding. With these mutations, the bacteria can still make proteins and survive in the presence of the antibiotic. They are resistant. The antibiotic treatment provides an example of natural selection in action. In the presence of antibiotics, susceptible bacteria die, but resistant bacteria have an advantage and multiply. The bacteria reproduce asexually and therefore make identical copies of the original cells. The populations of bacteria evolve, with the proportion of resistant bacteria greatly increased. Once established, resistance is hard to eliminate, and the resistance may spread to different species of bacteria. The easiest way to spreading resistance is when the resistance gene is located on a loop of DNA called a plasmid. We've already seen how resistance can occur when the antibiotic's target, such as a ribosome, changes. However, other antibiotics work by different mechanisms. In another type of resistance, a gene might code for a transport protein that pumps antibiotics out of the bacterial cell, preventing the antibiotics from harming the bacterium. In another type of resistance, a gene might code for an enzyme that breaks apart antibiotic molecules. The ability to resist an antibiotic can be gained by plasmid transfer. In a frightening yet realistic scenario, a relatively harmless bacterium transfers its resistance genes to a highly pathogenic species. What can we do to reduce the incidence of antibiotic resistance in bacteria? This brief list outlines important considerations.
All right, so the bottom line is antibiotics are ineffective against viruses. So over time, looking at the graph here, so this graph starts in 1980, and then the data does only go up to 2007, but this is for the number of new systemic antibiotic agents that has declined since 1980, and most of these drugs are in two classes called beta-lactams and quinolones. And so you can see here some of the different drugs and how they have decreased. So we have much less antibiotics to use. So we really need to do our due diligence and make sure that we are only taking antibiotics when we truly have a bacterial question. All right, so antibiotic resistance, this is evolution in action. This is, the big example is called MRSA, or methicillin resistant Staphylococcus aureus, which is a bacteria, and then methicillin susceptible. So if we are given an application of methicillin, the result is one of two things. If we have methicillin susceptible Staphylococcus aureus, then they die. If we have methicillin resistant Staphylococcus aureus, then they will survive. They will then reproduce, and then that resistant gene will proliferate. So you can see here a cartoon. We have our bacterium, we have our 3 variations, or in this case, we only have 2 variations. So this would be methicillin resistant, and this would be methicillin susceptible. We're given our antibiotic. These guys die, but these guys do not, and then they go on to grow the population that now will not work in the presence of methicillin. All right, so we just watched that video at the end. So our problem is that MRSA, methicillin resistant Staphylococcus aureus population increases. MRSA is the dominant strain, and then methicillin, as I said, is no longer effective. So we have to go back and revise evolution. What do we need to do to make sure that these are not the most prolific strains? And so these first came about in hospitals. That's where we first started to see them, and the problem is there was not any antibiotics that will work against them. All right, so how do antiviral drugs, that is what we need to take when we have a virus infection. So we have antiviral drugs are called interferons, and they stimulate the immune system to prevent replication of the invading virus. So some antivirals will target specific proteins, but other antivirals will target a specific stage of the virus life cycle, whether that's the attachment phase, the release of the genes into the host, the replication phase, the manufacture where you're making new viruses, or the release of the lytic cycle when those viruses are released. So when you have a virus infection and you're prescribed a specific antiviral drug for that pathogen, you really shouldn't use anything else because it is specific for that virus and that pathogen. All right, so back to our leukocytes, our white blood cells. So we've talked about our phagocytes, our vacuum cleaners briefly. They are going to help our other cells, our lymphocytes. So the bloody characters of specific immunity. You have something called a cytotoxic T lymphocyte. You have macrophages. You have helper Ts and B lymphocytes. All right, so this website, if you'd like to go to it, this is the website for the link. It has a lot of detailed information. If you were taking a microbiology course or if you were taking an immunity course, then this would be the level of detail that you need. So if you want to go through it and just check it out and see how they interact, you can, but you are not responsible for that level of material. So let's talk about these antigens and antibodies and how they work. So antigens, as I've already discussed, is a substance or molecule found on a cell or virus surface that causes antibody formation. So it's a protein on the surface, and it is characteristic of the cell surface or of the cell type. So different cells are going to have different antigens. The antibody is a globular protein that recognizes a specific antigen and binds to it as part of the immune response system. We've actually talked about antibodies and antigens before when we've talked about blood type and the ABO blood type relationship between antibodies and antigens. We just may not have referred to them as that. So if you look here, we have our A blood type that has our erythrocytes, our red blood cells have an A antigen. So in that blood, you naturally produce anti-B antibodies. So, and that's fine because they don't react together. You do not want your antibodies of your blood to react with your red blood cells. In the antigen B red blood cells, or the B blood cells have antigen B on their coat. And if you have that blood type, if you have B blood type, then your antibodies are going to be anti-A antibodies. Again, they're not going to react. So if you have an AB blood type, your blood cells produce the antigens both A and antigen B. And because the anti-B antibodies would react with the B antigens and the anti-A antibodies would react with the A antigens, you're actually not going to produce any or either of those antibodies in your plasma. And then O blood type has neither of the two antigens, does not have A antigens or B antigens, but because of that, they actually produce both the anti-A and anti-B antibodies. This is why it's really, really important that when you're getting blood that you get either O blood type, so that because it has no antigens, it's not going to react to any of the antibodies that might be present, or that you get a specific type that is good for your blood, that would match your blood type. So antibodies are specific to certain antigens. We talked about the similarity between the lock and the key of enzymes and substrates before. An immune system or immune response is triggered by non-self cells. So when your body matches or doesn't match with a cell type like bacteria, then it's going to trigger this immune response. So this is why transplants and blood transfusions, why it's so important to get this information correct. All right, so how do we produce antibodies now that we know how they work? So you have different types of lymphocytes that exist. And remember, we talked about a few different types on that slide, the B cell, the T cell, and the killer cell. And so each type is actually going to recognize a specific antigen. So that when the immune system is challenged, when we recognize that we have a non-self cell locating an invasion of a pathogen, the corresponding lymphocyte will respond. And so that lymphocyte will make clones of itself, and each of those clones will then have the antibodies for that pathogen. So this process is called clonal selection because the right lymphocyte was selected and then it was cloned. Some cloned cells, after the infection is taken care of, will remain in the body, a few number, and those are called memory cells. And so they are ready in case you get sick again. Here you have 3 different examples of B cells, so you can see they all have different antibodies on them, so they're going to react to different antigens. All right, so I'm going to give you some information about HIV. HIV virus attacks the immune system. It's called a retrovirus, which means it inserts its RNA into the host cell and then makes the host cell incorporate the RNA and turn it into DNA. And so when it does that, it then will attack our T helper cells. So they're a particular type of macrophage. You don't really need to understand their process. You need to understand that they are macrophages, and they are the specific cell that is attacked by the HIV virus, or the human immunodeficiency virus. And it attacks these T helper cells by binding to a specific protein on their surface. Once inside of the cell, the HIV RNA, as I said, is inserted into the macrophage's genome. Then it's going to trick the macrophage to doing transcription that includes the HIV's code, and then that's going to put out more viral RNA, and then later, subsequently, more HIV particles. So the immune system becomes weakened because we have less helper T cells or T helper cells, and we're also going to make
When HIV first enters the human bloodstream, the virus circulates throughout the body. The protective surface of HIV is studded with particular proteins called GP120, and inside is a capsid that contains the viral RNA and viral enzymes. Initially, the GP120 protein on the virus is only able to bind to a cell surface protein on the macrophage called the CD4 receptor. A second receptor protein, CCR5, must also be present on the macrophage for the virus to enter the cell by endocytosis. Viral RNA and enzymes are released into the cell's cytoplasm. Reverse transcriptase uses the viral RNA to synthesize first a strand of viral DNA and then the complementary DNA strand. The viral DNA then moves to the nucleus of the cell where it integrates into the host cell's DNA. Transcription of the DNA within the nucleus now results in the production of viral RNA. This viral RNA can serve as the genome for new viruses and can be used as messenger RNA to produce viral proteins during translation by the ribosomes. Complete viruses are assembled and released from the cell via exocytosis, which causes little harm to the macrophage. HIV cycles through macrophages over a period of years and continues to multiply while doing little apparent damage to the body. Eventually, the gene that codes for the GP120 protein is altered by mutation. The altered GP120 protein changes its co-receptor allegiance and now binds to a different co-receptor, CXCR4, which is found on the surface of CD4 plus T cells. The same processes occur within the T cells, resulting in new virus particles. However, as these new viruses leave the T cell, they rupture the plasma membrane, which kills the cell. As these newly released viruses invade and destroy other T cells, the body's immune response is weakened, and this leads directly to the onset of AIDS.
All right, so this is really what you need to take home about HIV. So you have your, this is really crazy, right? So you have two axes. First of all, you have your CD4 T lymphocyte count. So you have from 0 all the way up to 1,200 cells per millimeter cubed. And then over here, you have your HIV RNA copies per milliliter of plasma. So you have an infection. Okay, and starting with this infection, you have very little HIV. The problem is when we have an acute HIV symptom, we start to get more affected by our infection. And so you have a period where when you get infected, where the number of lymphocytes is going to decrease. And as your HIV copies is increasing, the number of lymphocytes is decreasing. Okay, so see, that's an inverse relationship. You'll have a drop as you have a slight rise. So you're fighting your infection. Okay, you're getting better. You're getting better. You're getting more lymphocytes. But then over a period of time, the HIV is going to start to win. You're going to start to have symptoms with HIV. And this is where you start to have those symptoms when your T lymphocytes hit what we call the AIDS critical level. So this is when your CD4 T lymphocyte counts are below 200 cells per millimeter cubed. So once your CD4 T lymphocytes get below that 200 cells, period, then you're going to start to have symptoms, and then we say that the patient's immune system is now ineffective, and now you have now you're not just dealing with the HIV particles, but now you have a syndrome that we refer to as AIDS, or the acquired immune deficiency syndrome. So the acquired immune deficiency syndrome is a result of the HIV virus, and it is because your body's T cell counts are so low that your immune system is deficient. So patients with AIDS are much more susceptible to just everyday infections, which could prove lethal. So you can see here, as we hit that level, any symptom, any infection is going to have issues for us, and our HIV copies are just going to continue to increase. So the life cycle of the HIV, I know we just saw it in the video, but just this is what you need to take away from it. The virus particle will infect the T cell. The protein coat will break down and it'll expose the viral DNA, RNA. The RNA is then converted to complementary DNA. The complementary DNA is inserted into the nucleus of the T cell. And then that T cell will start to produce HIV virus parts. And then the new HIV virus particles are released from the T cell. All right, so what are the risk factors in HIV transmission? Do you think you can put from no risk or lowest risk to highest risk? These terms, I'll give you a minute to pause the video and see what you can do. All right, I'm going to go ahead and go on. So your no risk, skin contact, toilet seats, mosquitoes. Lower risk, saliva or kissing. There is some risk, but ingestion, breastfeeding, and childbirth are going to be higher than saliva and kissing. Oral sex, sexual intercourse, and blood-to-blood contact are going to be blood-to-blood contact being your highest risk. So please be aware of that, and those are things you need to know for your risk factors of HIV transmission. And that is it for today. So make sure you bring your questions to class. We're going to do some in-class activities to really make sure that we all understand this information. Have a great day. See you in class.